Good morning, good afternoon, and good evening, everyone, and welcome to today's cutting edge conversation titled Discovering New Innovations in Oncology, sponsored by Optimer Group, BioIVT, and Chromatid. Thanks for joining us today. Our first speaker today is Dr. Cathie G. Miller. With a career that spans over 20 years at the bench, as well as director and global product manager, all involving the use of biospecimens in the fields of cancer, immunology, and virology, Cathie is now Senior Director of Product Marketing at Personalized Medicine at BioIVT. Our second speaker is Dr. David Bunka, Chief Technical Officer at Optimer Group. David has spent nearly 20 years developing nucleic acid aptamers against a wide variety of targets, including small molecules, disease-associated proteins, several cancer-associated cell lines, viruses, and tissue biopsies. This work has been facilitated through the use of high-throughput, automated aptamer selection methods. The last speaker today is Henry Sebesta, the Product Development and BD Manager from Chromatid Inc. Henry is a dedicated life sciences industry professional with years of experience in gene therapy product and business development. Together, our speakers will discuss how new technologies and innovations are being used to advance oncology research and treatment. Thank you all for joining me today. My name is Megan Loy, and I am the Senior Category Director for In Vivo Services at Scientist.com, as well as a board member of the North American 3Rs Collaborative. For the last 3.5 years, I've been working closely with our clients and suppliers to advance relevant categories, as well as offer animal welfare quality and compliance support. So cancer is a group of diseases that involves abnormal cell growth and the potential to invade or spread to other parts of the body. These contrast with benign tumors, which really don't spread. So it's basically a disease of uncontrolled cell division, and it develops and progresses in a series of changes and activities in the cell's regulators. So inhibitors of the cell cycle keep the cells from dying when these conditions aren't quite right. So too little of these inhibitors can actually promote cancer. It's basically letting something bad just keep reproducing in a nutshell. So cancer cells can actually multiply in culture, which is outside of the body in a dish, and without any growth factors, which is pretty amazing. And they don't even need protein signals to be added. So this is very different from normal cells, which need growth factors to grow in a culture. And cancer cells also ignore the signals that they get from their surroundings that usually would cause something to stop dividing. They completely ignore that. Cancer cells also fail to die when they're supposed to. So all cells undergo programmed cell death or apoptosis under conditions where they have things like DNA damage. Cancer cells ignore that. And emerging research shows that these cells might even undergo some metabolic changes that support increased cell growth and division. So not only are these cells not dying when they're supposed to, they're actually growing and dividing more. So why is this important? Well, oncology is the branch of medicine that deals with the prevention, diagnosis, and treatment of cancer, which is these cells that are growing without any limits. And it's really an interesting branch of medicine because it's interdisciplinary. So a lot of different experts need to come together for effective management and treatment. You might need a medical or surgical oncologist, radiologist, pathologist, and even cancer-specific experts or organ-specific experts that need to work together to manage treatment for individual patients. And that also means that the research needs to encompass many different areas and specialties. So there are hundreds of different groups dedicated to oncology research. And there's journals and advocacy organizations that work towards treatments and cures. So it's also collaborative. And actually this month, the month of May, is National Cancer Research Month. So this is the perfect time to discuss what the organizations represented today are doing in the realm of oncology research. And just a little bit of history for you. I thought this was fun. Our oldest description of cancer, although the word cancer wasn't what was used, it was discovered in Egypt, and it dates back to about 3000 BC. So it's part of a papyrus, and it's basically an old textbook on trauma surgery. And in that textbook, there are some cases of tumors or ulcers of breast tissue that were removed by cauterization. And they wrote about cancer that there really is no treatment. Moving along into the 1700s, somebody really did what is now routine, which is an autopsy. So this kind of first started coming back around, and it laid the foundation for a scientific study of oncology, or the study of cancer. And a famous Scottish surgeon during this time suggested that cancer might be cured by surgery, actually, which was kind of a newer or rediscovered treatment. And he wanted to decide, which cancers were operable or not. And he thought that if the tumor had not invaded nearby tissues, it was movable. So he said, go ahead and remove it. Well, coming along into the 19th century, we saw the birth of scientific oncology. So the use of the modern microscope really advanced our study of these diseased tissues. And there was now a scientific basis for pathologic study of cancer. So people started being able to link autopsy findings to the clinical course of illness, and then correlating that to the microscopic pathology. So now we're able to start linking these things together. And now in the last couple of decades, the growth in our knowledge of cancer biology has really led to remarkable progress in cancer prevention, detection, treatment. We know more things about our bodies and our genome now than we had previously, and it's an amazing advancement, but it doesn't change the fact that all this scientific knowledge is still based on things that are acquired and learned by hard work and discovery of all of our predecessors. And we know that there's still a lot more to learn. We could be doing a presentation like this in 20 years or in 100 years to talk about the latest advancements. So now that I've covered the last 5,000 years in about 30 seconds, I'm going to let our speakers tell you what's happening today. So with that said, I'd like to welcome David Bunka to the stage. David, thank you for joining us today. Feel free to take it away whenever you're ready.
Okay, so thank you for the introduction there. My name's David Bunka. I'm the Chief Technical Officer at Optimer Group, and I'm going to be talking to you today about our Optimer platform and how we can use that to deliver novel therapeutics for cancer treatments. So there's a lot of different treatments out there for cancer, and most commonly known are things like chemotherapeutics, which, as everyone knows, are really not very nice drugs at all. So if you can get a drug specifically to its site of action, it stops you having all of those off-target effects. So essentially, instead of having the drug floating around in your body, you can target it specifically to the cancer, to the tumor. You can get more of the drug to where it needs to be. Now, if you can do that, you can increase your drug's potency, you can potentially reduce your dose volume, you have a much safer drug because it's not going to be interacting with all of those other tissues that you don't want it to interact with, and that gives you a bit more flexibility around your drug as well. So there is a real goal in the development of novel chemotherapeutic agents to have them targeted, get them specifically to a site of action, and that's where the Optimer platform comes in. So just a bit of background on what an Optimer actually is. So an Optimer is a short synthetic nucleic acid molecule. So it's a piece of DNA or RNA that's been isolated through an in vitro evolutionary process to bind to a target of interest. So that could be, for example, a cancer cell. Now, there are a lot of other platforms out there that are being used for targeting various therapeutics. So you may have heard of antibody drug conjugates, for example. There are a few disadvantages of that platform that are overcome using Optimers. So first off, as you can see on the slide there, one of the main differences is the size of the molecules. So an antibody on average is around 150 kilodaltons. This compares to an Optimer, which can be anywhere from 5 to maybe 18 kilodaltons. So anywhere from a tenth to a 15th of the size. There's a number of advantages immediately from using that smaller targeting molecule. First off, obviously, is the ability of the Optimer to get to that tissue, to penetrate deeper into a tissue. So smaller molecules penetrate deeper into tissues than bigger ones. That's just simple science. But also there's an advantage in terms of the manufacturing of the Optimers. So as I mentioned, they're synthetic, so you can manufacture them on a much larger scale, much more reliably. Now, to come back to the point on size, I went to a lecture recently where they were talking about one of the problems with directed therapeutics is getting a physical mass of material into a cell. So if you have a large drug delivery vehicle like an antibody, your doses there can be as high as 6 grams in a patient. Now, you can imagine that's going to take a lot of time to get that amount of material into a patient, whereas something that's 1/10 to 1/15 of the size, that amount of material comes down quite dramatically. So there's a number of advantages to using a smaller molecule like an Optimer. Now, within our company, Optimer Group, we have different processes for different sorts of targets. So we can target small molecules, proteins, whole cells, and whole tissues with this platform. But what I'm going to focus on today is targeting cells and tissues. Now, as I mentioned, the Optimers are developed through an in vitro evolutionary process, and you can see that in the cartoon on the right-hand side of the slide. So we start here with our naive library. So this is a mix of trillions upon trillions of different sequences. So lots and lots of different sequences, all folding into different conformations. We incubate those with what we call a negative target, a counter target, represented by the little red cells in this case. So these are cells that you don't want the Optimers to recognize. So they could, for example, be healthy tissue in the case of cancer. You obviously don't want your drug delivered to healthy cells, so we want to remove any Optimer binders that stick to healthy cells. Anything that's left over is then recovered and exposed to the cells you are interested in. So in this case, that would be the cancer cells. In the little diagram, they're represented in green. Now here we want to recover the binders. So we collect the Optimers that have stuck to those cells, and then we amplify them up. So we recover that material, amplify it up, and essentially create an enriched library of binders that don't recognize the red cells, do recognize the green ones, and have an improved property. We then amplify those up to create a second generation and go round that cycle again. And each time we go round that cycle, we make it more and more difficult to bind to the positive target cells, the cancer cells, the ones in green, and make it more and more easy to bind to the negative cells. So what we're doing there is pushing the pressure to get the best binders to your target cells, the green cancer cells. Now, there's a couple of other things that we can do in that selection process. We can build in, for example, nuclease resistance into our library to make sure that the Optimers will survive inside the body. We can also build in steps to make sure that the binders get internalized by cells so that they take up the drug and it has a better chance of working. Okay, so this is, like I say, an evolutionary process for refining a population towards a cell type. Now, there's two different ways that we can actually go about this. The first one is what we call our hypothesis-free selection. This is where we don't really know what the receptor is on the surface of a cell that we're going to be targeting. We just know, for example, that we want to target a cancer cell and not bind to a healthy cell. So in the case in the cartoon at the top there, you can see the cancer cell on the left-hand side with a receptor represented by that little purple shape there, and the Optimers binding to that. So in that case, we don't know what the Optimers are going to be binding to. We just know that they're specific for the cancer type. Now, we can actually identify that biomarker later down the line through standard mass spectrometry and proteomic approaches. So it's hypothesis free because we don't know what we're targeting in the first instance, but we can identify it later. Now, if you do know what your receptor of interest is, we can actually take a different approach that we call target directed. Now, in this case, we start off with the recombinant version of whatever your receptor is. So let's pick a marker, CD4. We could take recombinant CD4 protein, attach it to a magnetic bead, and use that in the first few generations of that selection diagram I showed you previously. Now, that makes sure that the Optimer library that we're generating is targeting CD4. Now, in the later rounds, we then switch over to cells that express that protein on the surface. Now, this is really important because if some of that protein is, for example, buried in the cell membrane, we want to make sure that the Optimer binders are able to recognize the protein as it's expressed on the surface. So it's important to take this parallel approach. Protein first, cells expressing that protein second. And in that way, you get binders that target a specific receptor. So that's a bit of the theory on the process. Let's show some examples of this working. So in the example that's on the slide now, you can see across the top of the images on the top left-hand side of the slide, we generated some Optimer binders against a prostate cancer cell line model. And you can see in the third panel there, the Optimer binding very, very clearly and not binding to the control cell line that's running across the bottom there. And you can see from the merge that Optimer has actually bound to the cells, been internalized, and does appear to localize within the nucleus. Okay, so that's great. The Optimer binds, it binds specifically, and it gets into the cells. So can we attach a therapeutic to that? Well, we take a couple of different approaches to that as well. In the example here, we took a generic chemotherapeutic, in this case doxorubicin, which is not specifically targeted. And what we did here was took the Optimer that we'd already identified and created what we called a nanotrain tail. You can see that in the diagram, this little double-stranded region on the top right-hand side. Now we can load that double-stranded region up with doxorubicin because it's known to intercalate into DNA. So that's great. We've now got a delivery system in the Optimer with a tail that's loaded up with doxorubicin. And when we treated the cells, you can see the effect in the bottom left-hand side of the slide. So at sublethal doses of doxorubicin on the left-hand side of the graph, you can see there that the dox on its own has no real effect on either cell type. So that's great. In the middle, you can see that the Optimer is also having no effect on its own to either cell type. So the Optimer itself is not inherently toxic. However, when we create the Optimer with this drug-loaded tail, you can see that it has a very specific killing effect only to the target cells of interest. It doesn't touch the counter cell line, the healthy material. So you can see there what we've done is created a delivery system that takes a generic drug like doxorubicin and takes it only to the cells of interest. So it's targeted delivery of doxorubicin. We've also done this with other examples. So here we've generated an Optimer binder against the myeloma cell line H929, and you can see in the flow cytometry data on the left-hand side of the slide, the Optimer shown in green binds very clearly to H929, but doesn't bind to K562. And you can see the same in the fluorescence microscopy images. Across the top, you've got clear binding to H929 and no binding to K562. So we've got a specific Optimer binder. Now, in this case, that selection was done using our hypothesis-free method. So here, we don't actually know what receptor we are targeting. We just know it's specific to H929. And what we've done with that is then taken that Optimer and conjugated it to an antisense oligonucleotide therapeutic, an ASO. And what you can see on the graphs on the left-hand side there is specific delivery of that ASO using the Optimer. So the controls on the right-hand side, the untreated samples, obviously have no effect, which is great. What you can see in the pale blue line is that the Optimer drug conjugate has a very clear knockdown compared to the Optimer when it's treated with the K562 cells, which is the dark bar next to it. So again, you've got very specific delivery of this ASO knocking down a gene only in its target cell line. Now, the other important thing to note from that data on the left-hand side is if you look at the ASO on its own, looking at the purple bars and the dark blue bars respectively, what you can see there is the ASO on its own has a marginal effect to the H929 cells, but the ASO on its own actually does have a killing effect on K562. Now, if you compare that to the second bar, what you can see is the Optimer has actually stopped that killing effect from the ASO in K562. So again, we've taken an antisense oligonucleotide that was having an effect on K562 cells, but not on the H929s, and flipped that around. So we've made that ASO effective in H929 cells and stopped it having off-target effects in K562s. So again, we've made that therapeutic specific. We've also done this with small molecule therapeutics. You can see that in the graph on the right-hand side, showing dose response delivery of a novel chemotherapeutic agent to the cell line of interest there. So just coming towards the end of the presentation, I'd also like to look at how we can use Optimers as therapeutics in their own right. So in this case, we're not using the Optimer as a delivery vehicle, we're using the Optimer targeting a protein to have a direct therapeutic effect. So in this case, the Optimer is binding to a splicing factor that's mutated in leukemia. Okay, so what you can see in the micrographs on the bottom left-hand side there is the Optimer in red binding within the nucleus to the target protein. The target protein is actually being stained as well by an antibody, so that's the green. Now what you can see in that image is that the red and the green don't completely overlay. There are just specific loci where the Optimer is binding within that protein binding region. Now this is actually really important because not all of the protein in that nucleus is mutated. So what this is showing is that the Optimer is actually able to bind specifically to the mutated protein and not to the wild type. Now that's important because we need to be able to allow the wild type protein to have its effect. Now when we treat cells with this Optimer, you can see from the data in the middle of the slide there, the Optimer is able to partially restore wild type splicing by knocking out the aberrant splicing. So you can see that on the two gel lanes. In the untreated vehicle sample on the left-hand side, you see a very strong mutated RNA. So this is aberrantly spliced band on the left, whereas when the Optimer is treated, that aberrant splicing band is a lot fainter and the wild-type splicing band is a lot stronger. So we've restored that function. Now we're actually in the process of generating a second Optimer binder against these cell lines of interest to deliver that Optimer and again increase its efficacy. So hopefully I've given you a few examples of the benefits of the Optimer technology and of us as a company. So we've got a lot of experience developing Optimer binders against all sorts of cell types. We can do this very, very quickly, so this gives us faster time to market. The Optimers, as I mentioned, are entirely synthetic, so we can create them and manufacture them on much larger scales. And they're very easy to conjugate other things to. So I've shown you a couple of examples of chemotherapeutics or other oligonucleotide-based therapies. This is all routine chemistry to attach those two molecules together. They're very specific, and the Optimers are themselves, if they're in solution and can be kept either frozen or kept clean, they do have a very long shelf life as well. So you don't have drug molecules going to waste. So that's the end of my presentation. I hope you found it interesting. And if anybody has any questions, I'd be welcome to hear them and try and answer. Thank you for your time.
Thank you so much, David, for your presentation. I'd like to now welcome Cathie Miller to the stage to begin the next presentation. Welcome, Cathie. The floor is yours when you're ready.
Thank you, Megan. As Megan said earlier, I'm Dr. Cathie Miller, and I'm the Senior Director of Marketing for Personalized Medicines at BioIVT. Mutation analysis is an important research and diagnostic tool for the studying of cancer and other diseases. Combining our expertise in human tissue procurement, specimen selection, sample preparation, and project management, BioIVT offers both standard and custom services for extraction, library preparation, sequencing, and bioinformatics. Today I will discuss how BioIVT, your complete biospecimen provider, goes further with in-house NGS solutions. BioIVT has brought together organizations across the biospecimen spectrum to provide you a single source for your biological matrix needs. from different human and animal matrices for controls and surrogate matrices, to disease state and match normal controls for liquid biopsy, leukopaks and immune cells for cell and gene therapy development and validation, as well as a cohort of over 1 million cases with H&E images, confirmation of diagnosis, and over 200 clinical and specimen-specific data points to integrate into machine learning development, we have the biospecimens you require. We are a global organization, excuse me, with 18 operating locations, over 3,500 IRB-approved collection sites located around the world. Our sites span a range of clinical operations from local doctors' offices to large health systems to 9 BioIVT-owned donor centers. And BioIVT knows that quality, ethically sourced biospecimens are essential to your research. Thus, we have set the standards for ethical collections with additional levels of certification, accreditations, and approvals, including ISO 9001, CAP, HTA, CLIA, and FDA as appropriate. BioIVT provides products across the spectrum of human and animal-derived specimens, including both normal and disease state biological matrices, various primary immune cell and immortalized cells, as well as our ADME-Tox-related products and services. Our experienced scientists and technicians ensure fit-for-purpose solutions to accelerate your development, from customized processing to creation of slides and TMAs, to cohort mutation screening, to high-quality bulk plasma offerings. We get what you need in the format you require. Our products readily fit into workflows for discovery research, cell biology, immunology, diagnostics, and bioanalytics or biomarker research. One of the strengths is our Astrian Human Biorepository with its CAP accreditation and ISO 9001 certification. For over 30 years, this biorepository has been providing researchers across pharma, biotech, and diagnostic industries with ethically procured high-quality tissue blocks that have gone through rigid quality control processes, including board-certified pathology confirmation of diagnosis, independent review and verification of clinical data and tumor staging, as well as additional services to ensure fit-for-use solutions for our clients. BioIVT's Astrian Human Tissue Repository has been collecting biospecimens to our best-in-class standards, delivering consistent quality for our tissue specimens that fit well with NGS requirements. Over the years, we have had clients come to us and want to screen specific cohorts within our biorepository. We have always been happy to partner with our clients to ensure their success, and so with each project, we applied our gained knowledge to other projects, adopting standard screening practices to expand our offering. Currently, we have over 8,000 screened specimens in the bank ready to ship today. And after years of cohort selection followed by sample and library prep, we undertook to determine success as observed with our inventory and processes. We presented these results at the Molecular Medicine Tri-Con in 2019. Interestingly, and contrary to many of our clients' perceptions, we were able to show that proper upstream procurement and processing, as well as quality control, led to success, which was not affected by, most surprisingly, age of the block. And in 2019, to better service our clients, we invested in bringing NGS technology in-house. Our process is that clients will consult with our specialists regarding the desired cohort and mutation of interest, exclusions and inclusions, incidence rates for general population and in BioIVT's bank, if known, and the testing process that is required. Sometimes this can mean a novel test development, replication of a commercially available assay, or any available assay that works. Once the desired cohort has been identified, BioIVT search engineers work with our project specialists to identify available inventory ready to be screened, as well as additional sources for the most appropriate collections if additional specimens are required. At BioIVT, we plan and procure additional specimens before needing them to ensure that the project continues to move forward smoothly and on time. Based on our current inventory and known historic accrual rates, we can accurately estimate the timeline for the projects. BioIVT has over 10 years of expert sample and library prep experience, working with key industry providers to troubleshoot their processes and kits to ensure success. And because we've been doing this for over 10 years, we have verified partners that we can work with for any required assay type. As stated above before, in 2019, we validated NGS in-house with an Illumina NextSeq 5500 and an Illumina iSeq, adding this to this soon after 3 Thermo Fisher Genexus integrated sequencers. We have been processing around 800 samples a month since early 2020, both for specific client projects and to add to our ready-to-ship inventory. Once the cohorts have been collected, sample library prep prepared, and the runs are finished, it's time for data and tissue delivery. Products can consist of data alone, data and tissue blocks, processed tissues into blocks, slides, or TMAs, as well as additional features such as pathology consultation, markup, and more. Validated assays in-house include the Oncomine Precision and Comprehensive assays, the AlloSeq TX17 HLA assay, and the AmpliSeq BRCA and Comprehensive assays. BioIVT is engaged in multiple successful projects with our clients, and here I summarize 3 such projects. These projects include a large diverse screenings of diverse cohorts to deliver a wide breadth of mutation-positive specimens, to a very specific assay looking for a specific gene mutation within a highly defined cohort, to continuous HLA typing of specimens. For each of these projects, our molecular team has met with the clients, determined their need and the best solution for their need, and implemented a successful plan to identify the right cohort of samples, determine the right platform, assay, and isolation solution, as well as perform data analysis and deliver results to the client in the data format required. If you want to see what's available to ship today, visit bioivt.com and search our available inventory. Pick your own inclusion and exclusion criteria to refine your mutation cohort to your needs, or sign up for our monthly alert available of all available screened specimens. Also on our new BioIVT digital portal are resources to help support your research, from white papers to webinars to blogs, to documentation, quality assurance information, and more. BioIVT partners with you to elevate your science. And with our concierge disease state specialists, we focus on your needs. Reach out to them directly to ask questions, determine if a collection is viable, find alternative solutions, or to pull in our team of in-house subject matter experts to help make sure you get the cohort you need. And it's not just our ability to service you with the right cohort of specimens with the right mutation data that makes us unique. Our wide access to donors worldwide provides sources for our extensive ready-to-ship inventory and allow our ability to customize your collection quickly, efficiently, and effectively. Our high ethical and quality standards are industry-leading and important to the core of who BioIVT is. Our responsive customer service team, including our business development members, customer service, lab operations, and logistics, work together to provide top-notch, seamless solutions to our clients. Thank you.
Thank you, Cathie, for your presentation. I'd now like to welcome Henry Sebesta to the stage to begin the next presentation. Welcome, Henry. The floor is yours whenever you're ready.
Okay. Hi, everyone. My name is Henry Sebesta, Product and Business Development Manager for Chromatid. I've been with Chromatid for a little over 4 years, and I've gotten the chance to do some of the product development work for a few of the assays I'm going to be discussing today. So I thank all of you for joining me, and I thank scientists for hosting this. Today, I'll be walking through how Chromatid's proprietary cytogenetic analytical platforms, directional genomic hybridization, and pinpoint FISH can be used as in vitro research tools to assess the genomic impact of gene and cell therapies for oncology. First, a bit of context for those who may not be familiar with us. Chromatid is a cytogenetic analysis company based just outside of Boulder, Colorado. At the moment, we're a team of about 30, but we've been rapidly expanding over the past year or so, finishing up a B round of financing at the end of 2021. We were initially a spin-off of a radiation biology lab out of Colorado State University, funded by NASA as a way to monitor the effects of ionizing deep space radiation on their astronauts. We still do some of that work, but our primary focus these days is on helping gene and cell therapy developers bolster their regulatory data filings with unique single-cell analysis that combines the innovative aspects of our technology with the validated standards of regulatory requirements. You can see here from a couple of studies with our first customers in the space, Intellia Therapeutics and Editas Medicines, where we help them identify and track translocation rates in their treated cells. We do this by using a technique I've already alluded to called directional genomic hybridization. This technique is similar to FISH, but uses our proprietary bioinformatic design and cell prep methods to selectively strip daughter strands during cell replication. This leaves a single unidirectional chromatid on either side of the centromere in metaphase chromosomes that, when we hybridize our single-stranded oligo probes to. The result is a hide-based assay that has orders of magnitude better resolution than most FISH probes, and also captures every class of genomic structural rearrangement, including translocations, copy number variants, inversions, and sister chromatid exchanges. There are two iterations of this technique that we differentiate primarily by the amount of genomic coverage of the probed area. DGH Insight on the left here is our localized version of the assay with a resolution down to about 2 to 3 kb. It's mostly used to track edit sites or characterize transgene integration events associated with editing or viral delivery. DGH Screen on the right is our unbiased approach where we paint every chromosome with probes so we can identify down to a specific chromosomal arm where rearrangement events are happening throughout the genome. Screen can be used for a variety of applications ranging from genomic stability assessments for cell line characterization to identifying off-target or unintended genomic effects of any therapeutic modality. You can see a couple of real-world examples of Insight and Screen here. On the left, what you're looking at are Insight probes in one metaphase spread of a dual edit system in hex cells. Those circles there are highlighting the green and red probes spanning 2 edit sites on 3 homologs. There are 2 examples of a normal edit site where the red and green probes are on the same side, and an example of an inversion here where part of the red probed region between the edit sites flipped over to the opposite chromosome. On the right, we have our prototypical karyogram for Screen. This is meant to demonstrate Screen's ability to detect various rearrangements throughout the cell and how, using different labeling patterns and chromosomal morphology, we can identify which arms of the chromosomes are occurring. Before we get into some data, I wanted to highlight some of the concerns around current popular analytical methods. The World Health Organization and the National Institute for Biological Standards and Control put together a collaborative study to try and establish a standard for lentiviral integration analysis. What they found was that there was some variability between the 31 labs that participated and between the 3 PCR-based methods used when trying to determine lentiviral copy numbers per cell. I've included an excerpt from the study here, which I think summarizes the overall issue nicely. The bottom line is that while PCR data is critical, there are limitations around amplification biases, reaction conditions, and data interpretation. So the global gene therapy community is still on the lookout for analytical methods that can help bridge the gap. Here's an outline of a study we did that's representative of how we propose to shine some light on this issue. You can see here ideograms of the study we set up with a collaborator that was using an unidentified viral vector to deliver an integrational transgene to what they thought was a targeted site on chromosome 22. For this study, we made probes for their target site in green, their integrant in yellow, and we added paints on chromosomes 1, 2, and 3 in the background as proxies for monitoring genome-wide off-target effects. Here's an example of a cell from their control human dermal fibroblast line without their integrant. You can see the green probes there bracketing their target integration site, but we wouldn't expect to see any integration here, so all looks normal. What we found in the rest of their iPSC samples, however, was not normal and was a bit unexpected for both of us. We found that after looking at about 600 cells across their samples, only 6% of those cells had integrations that were only at the target site. A good portion, about 40%, had both on and off-target integrations, and almost half of their cells had no on-target integration whatsoever. You can imagine they were pretty surprised as they were expecting a much cleaner system. So we went on to analyze another cohort of about 200 cells, gathering the same data but with a slight twist. To this cohort, we added a control probe on chromosome 8 to try and help us quantify the copy number at each integration site. The control probe on chromosome 8 was of a known genomic size and fluorescence intensity, and by using that and the known genomics of our insert probe, we were able to calibrate our other signals against it to give an estimate for the number of inserts at each integration site. In addition to even more drastic off-target data observed in the second cohort, we were also able to glean some pretty interesting copy number data. You can see the number of integration events on the y-axis here and the integrational copy number on the x-axis. This data indicated that the vast majority of these inserts, both on and off target, were being integrated as only one copy, although we did see a fair amount of variance, including 9 integrations with over 5 copies. So, to summarize, we were able to provide our collaborator with not only a cell-by-cell distribution of the number of integration events, but an event-by-event analysis of the number of inserts at each integration site. Shifting gears a bit here, I'll quickly walk us through another case study to demonstrate potential applications for another iteration of our technology, DGH screen. This study was performed with the NIST Genome Editing Consortium, and we are analyzing the genome in a bottle progenitor cell line provided by the Coriell Institute. In addition to being the most sequenced cell line in the world, as they say, Coriell had already performed G-banding analysis and found a potential inversion on 3q. So the goal of the study was to use DGH to confirm previously observed rearrangements and to see if we could find anything that might have been missed. You can see here a good example of one metaphase spread from these cells that shows a variety of rearrangements were present throughout the genome. As you can see, many of these were large structural rearrangements like translocations and inversions that were difficult to detect using the previously described deep sequencing methods. Without getting into a detailed breakout of the prevalence of each rearrangement, we were able to confirm the suspected inversion on chromosome 3, and we also found quite a few new inversions and unexpected rearrangement complexity. Here are a few examples of some of those newly discovered inversions and the recurring complexity associated with chromosome 16 on the left there. In addition to that multiradial structure that we observed in 4% of cells, there were other hallmarks of instability, including decondensed or elongated centromeres that we were able to confirm with standard karyotyping methods. Here's an example image from our G-banding validation on the same cells. Much of the instability and rearrangement data on chromosome 16 matched our DGH results, as you can see with that elongated centromere right there in the middle. But as expected, there were many rearrangements, including the previously known and newly discovered inversions that we weren't able to see with G-banding, which kind of highlights the power of screen's kilobase level resolution over the megabase level resolution of the current regulatory karyotyping standard, G-banding. So with that, we've walked through a couple high-level applications of how our DGH platform, but I want to briefly touch on the last iteration of our technology, pinpoint fish. The oligos we use for our DGH probes can also be used as extremely high-resolution, single-stranded fish probes, which we can then ship to you as kits or run in our lab as a custom assay service. Our bioinformatic design enables researchers to run 2 to 3 kb resolution fish assays as opposed to the approximate 100 kb lower limit of back-based fish, which can be run in your lab using standard fluorescence microscopy equipment. We also recently launched our first standard fish kits, which include a variety of sub-step and sub-telo probes, as well as our TP53 and SEP17 gene kit, all of which range from about $15 to $18 per assay. You can learn more about our fish kits through our website or by reaching out to sales@chromatid.com. So we've gotten a chance to cover a variety of applications for our technology, including our methods for edit site tracking and integrant profiling with DGH Insight, unbiased whole genome stability and rearrangement assessments with DGH Screen, and our ultra-high resolution fish kits and assays. This workflow provided here is an example of one of our service projects, but as I've mentioned, we're also happy to ship you out a kit and let you run these assays from the comfort of your own lab. At the moment, many of our customers are combining Insight, Screen, and G-banding for the ultimate cytogenetic analytical suite that provides targeted, unbiased, and standard validation for their regulatory filings. So far, we've contributed data to 4 different filings from 4 different companies, with a 5th on the way, and we hope we can continue to increase that number with a few of you. If you're interested or you think you have a potentially relevant project, we'd be happy to discuss it with you. Please feel free to reach out to the contact info here or my personal contact info provided here. Thanks again to everyone for your attendance. I really appreciate your time and attention, and thank you for scientists for giving us the opportunity to present.
Thank you, Henry. That concludes the presentation portion of this event. So we'll now move into the Q&A. And as a reminder, if you have a question, you can submit it using the Ask a Question panel that's shaking on the screen here. Okay, let's jump in. It looks like we have quite a few questions here. This is really exciting. Oh, excuse me. Let me just start to bring some up here and we'll get going. Okay, great. So it looks like we have a wonderful first question. So is BioIVT able to detect mutations using liquid biopsy?
Hi, Megan. Yes, we are actually in the validation process of obtaining cell-free DNA using different isolation methods to validate looking at mutations for liquid biopsy. So that's in development as we speak.
Excellent, thank you. Another question for Dr. Bunka. A really interesting talk, thank you. You showed some data about testing the specificity of these vehicles against cancer versus healthy cell lines, but obviously there is a lot of heterogeneity of cell types and protein receptor expression in the human body, particularly when it comes to the hypothesis-free targeting. How do you ensure there aren't off-target effects in other diverse cell types outside of more homogeneous cell lines?
Yeah, that's a very good question. It actually comes down to how you design your selection approach. So every Aptimer selection program is different. And we actually spend a lot of time talking to our clients about the sort of off-target effects that you might experience in a real-world situation. So we try and build in as many of those off-target cell lines as possible. So not just targeting cancer versus healthy in, let's say, the pancreas, but you might want to look at other sort of epithelial cells, for example, if you were going to be injecting it. So it's all about getting that cell line right. And we can use multiple cell lines if we want to. Now, there is an addition that we can do as part of the selection process is what's referred to as in vivo selection, where we actually take an animal model, for example, and put the whole Aptimer library into that model. So in that situation, the Aptimers are going to be exposed to every sort of tissue type as they circulate around the body. And then when we recover the tumor, we'll obviously be recovering the Aptimers that have survived that circulation around the body and made it to that tumor. It's a bit more involved. It's a more lengthy process, but it's something that we're very interested in doing. I hope that answers the question.
Thank you. This one's for Cathie. What QC acceptance criteria are used to determine if the DNA and RNA extracted from an FFPE block are suitable for NGS screening?
That's a great question. We actually go through a number of different validations. So we use quantification using qubit for DNA high sensitivity as well as RNA broad range assays. We use the RIN number as well as the DIN functional range for DNA and the base pair sizing range, all from Agilent Technologies. And then we also do DV200 analysis. However, we like to make sure our clients are aware that this data is really dependent upon the isolation method and the technique that is used, which both of those can have an impact on the RNA fragmentation. So it's very important to look at that.
Thank you. This next one is for Henry. What type of cells do you require for DGH analysis?
Yeah, so DGH can be done on any cells that you can get metaphase spread out of. The DGH portion does require actively dividing cells, and so some of the most common cells we work with these days are iPSCs, T cells, NK cells, and then we accept live or cryopreserved samples. You can use your culture methods, or we can help you develop.
And how many cells do you need to look at for DGH analysis?
Well, the number of cells we look at is going to vary depending on the prevalence of the event you're looking for. And so the less frequently you expect to see the event, the more cells we're going to need to count for statistical power. So if you're looking for edit site rearrangements, for example, we'll probably have to count more cells than if we were screening clones. But we typically fall in the 200 to 600 cells counted per sample range for edit and integrant tracking, and then maybe in the 20 to 100 range for the whole genome.
Thank you. The next one it looks like is for David. Can Optimer binders be used in cancer applications other than therapeutics?
Yeah, absolutely. So we're actually using our Optimer binders in all sorts of applications. So not only therapeutics, but diagnostics, research tools. Essentially anywhere that you could use a binder, you can use an Optimer. So you might, for example, in your studies, use flow cytometry. So we can generate an Optimer binder that's specific to a cell type, pop a fluorophore of choice on there, and then use that as a flow cytometry reagent or a FACS reagent, so you can actually sort and recover your cells. And as I said, you can also use them potentially as diagnostics as well. So there's a lot of examples out in the literature where people have conjugated all sorts of tags, including radioisotopes, for example, to allow the Optimers to be used as tracer molecules to look at sort of tissue localization, cancer sites, that sort of thing. So yeah, all sorts of applications. It's not just therapeutics.
And do you select for Optimer binders with endosomal escape properties?
Very good question. It's not something we specifically select for, but as you could see in some of the examples that I showed in the presentation, there is evidence that the Optimers are binding to cells, getting internalized, and then escaping those endosomes and making it into the nucleus. So, we don't specifically select for that, but there is evidence that it happens anyway.
Excellent. And one more for you. If you select for cell phenotype rather than a specific biomarker, how can you discover the specific receptor that the Optimer targets?
Yeah, again, that's a very good question. You're quite right. Any selections that go through our hypothesis-free approach, at some point you're going to need to work out what that biomarker actually is. So this is actually becoming more routine practice. So you can actually use a binder molecule to pull out a biomarker, so attach it to a solid support like a magnetic bead, for example, and use that to bind to the surface of a cell, so stick to its receptor of interest, lyse those cells, and then pull that biomarker out and send it for example, tryptic digest and mass spectrometry. So there are methods out there for identifying those biomarkers once you have a binder.
Wonderful, thank you. Henry, it looks like we have a couple more questions for you. The first one is, can you only work with human cells?
No, we can work with anything with a sequenced genome that we can design probes against, including research models like mouse, pig, or non-human primates.
And then for the DGH analysis, do you only run that as a service?
No, actually, like we touched on a little bit at the end there, we also sell our custom and standard probes as kits. And so any of these assays can be run in your lab. You'll just need a fluorescence microscope with the appropriate filters. Our cell prep method and hybridization protocols are pretty simple, but we also offer on-site or team support for those who would feel more comfortable with us walking through it.
Thank you. And then we have a couple questions for Cathie. Is BioIVT able to detect mutations using liquid biopsy?
Yeah, so we're validating that process right now with cell-free DNA, and we have some initial promising results.
Excellent. And if you've used BioIVT for NGS mutation data screening, are you able to have access to the raw data files?
Yes, actually, we share that as a matter of principle. So we have the ability to share the FASTQ, the SAM, the BAM, and the VCF using cloud-based storage solutions based on the client's needs.
Excellent. And it looks like this next question is for David. What is the size limit of molecules that can be conjugated to Optimers?
That's a very good question. I'm not actually sure the answer to that. I know that we've conjugated on site here everything from small molecules, which are down to about 1.5 kilodaltons, I think was the smallest ones that we've conjugated, all the way up to double-stranded siRNAs. So they are in the region of sort of 30-ish kilodaltons. But we've seen examples where people have conjugated whole liposomes, for example, or even viruses. So you can conjugate things that are up to megadaltons if you need to.
Excellent. And this one looks like it's for you as well. Do Optimers induce immune responses?
Oh, that's a very good question. That's one I get asked all the time, actually. They can, but most often they don't. So it really depends on the sequence of the Optimer and the modifications that are held in its backbone. So it's well known, for example, that particularly GC-rich sequences can stimulate toll-like receptors, so you get complement activation, for example. But for the most part, if that happens with your Optimer, you can actually engineer those sequences out, so reduce the GC content of your Optimer to limit that effect. But for the most part, they don't stimulate the immune response. It's only a very few sequences that do.
And another question for you. What are the PK or PD properties of Optimers in vivo? This is reminiscent of the nanoparticle story where targeting looked great, but in practice they were taken up by liver macrophages. Any data on systemic delivery of Optimers in vivo?
That's a very good question as well. Yeah, so it's very true that Optimers, like any other nucleic acid or a lot of other drugs, they do accumulate in the liver, in the kidneys, mainly because that's their route out of the body. But there are a lot of studies that have been done with subcutaneous and IV injections of Optimers. And what they quite often show is that the Optimers circulate around the body, localize in the tissue of interest, so where they've been designed to go, and then you see a pool of them in the liver and kidney as they're excreted. So you do see tissue localization as well as accumulation in the liver and the kidney.
Excellent. Thank you so much. Another question, does your technology require cell clones?
Is that for Optimer, sorry?
You know what? That's a good question that you have. I'm not sure who that question was for. Okay. Well, with that, little bit of a confusing question. Thanks so much for joining us for today's presentation, and a big thanks to Optimer Group, BioIVT, and Chromatid for sponsoring this cutting edge conversation. We now invite you to take a moment to share your feedback on the survey, and we'd really appreciate your input. If you still have a question, or if we didn't ask your question to the wrong person, it's not too late. Feel free to send it in now, and we will forward it along to the speakers following the event. If you're interested in learning more about Optimer Group, BioIVT, and Chromatid, we invite you to check out the resources panel. And if you're interested in seeing what's coming up next in the Cutting Edge Conversations series, you can click the register button on the slide to find out more. In closing, we hope you enjoyed this Scientist.com webinar, and we'll see you next time.